Hello and thank you for stopping by my poster. My name is Mathias Kalxdorf and I'm working as a computational biologist at GSK located at the EMBL campus in Heidelberg, Germany. Today I will give you a brief introduction to my poster, which I will present in person at the ISMB and ECCB 2023 in Lyon, France. The title of my poster is Inference of Mechanistic Insights from Multi-omics Data Facilitating the Drug Discovery Process. Let's start with a very brief introduction summarizing the motivation for our work. The drug development process is typically costly and challenging, particularly due to the high failure rates at the late stages of the drug discovery process. This issue can be attributed to various factors, with a prominent one being the lack of comprehensive understanding regarding the disease mechanism and the causal effects that arise from perturbing selected drug targets. However, in recent years, the availability of multi-omics data increased a lot. This wealth of data encompasses various molecular-level omics information, such as transcriptomics, proteomics, and more. The integration and analysis of the vast amount of data offer opportunities to address the aforementioned knowledge gaps in drug development. Here we present MIMO, mechanistic inference from multi-omics data. It applies two fundamental steps to infer mechanisms. First, it overlays observed multi-omics data with prior knowledge causal networks, for example as available from the OmniPath database. And second, it adds measures of confidence, like scores and p-values, to mechanistic hypotheses by joining confidence from causal reasoning, from multi-omics-based protein activity estimations, and from the underlying prior knowledge. We benchmarked and compared MIMO against published tools, including Carnival and SAMgraph, based on chemical and genetic perturbation datasets. MIMO generally showed outstanding performance, and I would like to share here now some of the results in more detail. But let's first start with the fundamentals of MIMO, the applied scoring algorithm as illustrated in Figure 1. MIMO identifies and scores causal mechanisms underlying multi-omics phenotypes by applying a unique scoring algorithm which integrates confidence levels of node regulations across observed omics layers with confidence levels of causal protein interactions in the used prior knowledge network. The resulting mechanistic hypothesis scores are normalized between 0 and 1. The capability to A. integrate information across omics layers, to B. consider different edge confidence levels of the used prior knowledge causal network, and to C. report mechanistic hypothesis scores makes MIMO unique in comparison to other available tools like Carnival and SAMgraph. An exemplary comparison of the tools is shown in Figure 2. Carnival is capable to handle multi-omics input like MIMO, however it is not reporting mechanistic hypothesis scores, which hampers spotting the true mechanistic paths among all possible hypotheses. SAMgraph in turn reports confidence metrics per hypothesis, however it can only handle a single omics input at a time and cannot consider confidence levels for respective edges in the used causal network. In the here shown example, only MIMO enables spotting the true mechanisms from the multi-omics input. To showcase the outstanding performance of MIMO, we show in Figure 3 the results of the three tools in a benchmark dataset. It is important to note that there is generally no mechanistic ground truth knowledge available. Hence, the performance of the tools had to be measured indirectly. For that purpose, the following four performance parameters were evaluated. First, how many of the observed phenotypic effects can be explained by significant mechanistic hypotheses. Second, how correlated are the determined hypothesis scores with literature text mining evidence for respective mechanistic paths. Third, how consistent are hypothesis scores with Carnival results, as Carnival is generally a robust causal reasoning pipeline, just lacking the capability to additionally report confidence measures for the hypotheses. And four, how consistent are hypotheses and their scores across multiple individual biological replicates of the same experiment, which shows the well-studied cellular system of M1 polarized macrophages as the experimental basis. In these cells, STAT1 is a key transcription factor which mediates and maintains the polarization, and it is known that inhibition of STAT1 in M1 macrophages leads to the suppression of the pro-inflammatory phenotype. Here, we measure the effect of STAT1 knockout using CRISPR on proteome, phosphoprotein, and transcriptome, and inferred causal mechanistic paths linking STAT1 perturbation to observed differentially regulated proteins and genes. The results indicated that 1. MIMO can generally explain the highest fraction of differentially regulated proteins and genes with significant mechanistic hypotheses. 2. Its hypothesis scores are well correlated with evidence scores from literature text mining. 3. MIMO is generally highly consistent with Carnival, however is capable of additionally reporting hypothesis confidence levels. And 4. is reporting highly consistent hypothesis across multiple individual biological repeats. These outcomes prompted us to evaluate the results in more detail. In Figure 4, we show examples of additional insights which can be gained from MIMO results. First, observed differentially regulated proteins and genes can be clustered by mechanisms. Results indicate here that there are multiple mechanistic clusters which explain the phenotypic effects induced by the STAT1 knockout. We can further evaluate for each cluster which nodes along the underlying mechanistic paths are driving the separation. In case of cluster 6, used here as an example, we can see that these effects are all driven by inhibition of alpha interferons like IFNA1, IFNA2 or IFNA10. Second, MIMO can be used to unbiasedly identify mechanistic causal origins of observed multi-omics phenotypes. Here we chose 2 CRISPR knockouts and 1 cellular stimulation as examples. In case of STAT1 knockout in M1 macrophages, MIMO ranked STAT1 among the top 5 possible causal origins for the observed phenotypic effects. In case of STAT6 knockout in M2 macrophages, MIMO ranked STAT6 among the top 3 causal origins, and in case of stimulation towards M2 macrophages by interleukin-4, MIMO ranked IL-4 among the top 2 causal origins. So in conclusion, MIMO enables robust identification of molecular causal mechanisms, explaining phenotypic effects by integrating prior knowledge networks with experimental multi-omics data. Its novel scoring algorithm allows ranking of mechanistic hypotheses based on data-driven confidence metrics, and in benchmarks, MIMO performed favorable in comparison to other tools such as Carnival and SemGraph. As shown based on 2 examples, MIMO can provide additional insights into biological systems, and we foresee that MIMO will impact the drug discovery process by providing novel insights into disease mechanisms as well as unraveling causal effects induced by perturbing select drug targets, allowing early identification of potential unfavorable side effects. In case of questions, comments, ideas, please feel free to reach out via email or get in touch with me at the ISMB and ECCB in Lyon. Thank you for your attention.